Can you hear me all right? I am delighted to be here, and I am really honored to be invited by Helen Heslop. I don't know if I told you this, but your team has really been my idols through much of this. Helen, working with Malcolm Brenner and Cleo Rooney, really have, I think, been an exemplary team that has guided this field from the very beginning. So let me get applause for Helen. So, as Helen mentioned, here's my disclosure slide. I'm going to give you, yeah, it's sort of funny these days. I'm going to attempt to give you a completely new story today. I always try to focus on unpublished work, but today I'm going to give you an entirely new story that we've come up with. And so I invite you to criticize it afterwards, and I'm going to try to leave enough time for that. A title is The Hunger Games, How Starvation Limits Aging and Enhances Anti-Tumor T Cells. An alternative title could be How Physiologically High Extracellular Potassium Affects T Cell Memory and Effector Functions through Epigenetics. So let's see if we can get through this. And so what I'll be talking about today are situations where there's a lot of extracellular potassium. So for physicians in the room, you know you can't have in your blood more than about 5 millimoles of potassium, and very, very little. But there are situations where we've measured very high extracellular potassium, including within the stasis of a tumor with necrosis and apoptosis. And we think it happens in other situations that were evolutionarily important. So anytime these bags of cells containing lots of potassium, and I'll explain how much potassium there is, when they break down, like during frostbite or on a crush injury or in tumor, they release potassium. And so you can have locally high levels of potassium. And so what's the impact of that? So, but let me take a step back, because I think this room is really educated in the field of cell and gene therapy. But let me review for a second why we love cells. Why have we been trying to, focus on giving cells as treatment for cancer for 30 years or more? Well, first it's a great research project. We can identify and enrich for the exact cell population and the qualities of cells that are associated with effective anti-tumor responses. We can also, because we have the cells in a dish, we can pharmacologically or genetically modify those cells to give them the right qualities that we're looking for. And we can add or edit genes to alter their functions and also give them any specificity we can engineer. And you can see that works in the 2 approved CAR-based therapies this year. We can also give unlimited numbers, so we often give up to 10 to the 11th lymphocytes to patients. But here's a charming thing. I think we can lymphodeplete, so we don't have to, we can take the host who has a corrupt tumor microenvironment, and because we have the lymphocytes in a dish, we can ablate the patient and kill off all those myeloid-derived suppressor cells and Tregs temporarily, and we can lymphodeplete very effectively. So how do we do this? So the very easy general schema of growing naturally occurring lymphocytes simply involves excising a tumor from a patient, expanding, adding interleukin-2 after you chop up little pieces of that, picking the wells that are positive and giving them back. It's very straightforward. Of course, now we have gene engineering, which in a way is even more straightforward. We just take lymphocytes from the peripheral blood and give them back. And so the thing we face is that the tumor, there's immunosuppression, and it's very complicated. But most of these mechanisms that have been worked out don't address another fundamental question about tumor infiltrating lymphocytes I want to describe today. Okay, so the central paradox for us is, why are tumor infiltrating lymphocytes dysfunctional? So we know that, all of us know that, right? If you resect a patient's tumor and you look at it, there are lymphocytes sitting right next to a tumor doing nothing. But at the same time, those lymphocytes can grow exponentially and exhibit what we are calling stem-like behavior. And we've shown that it really is stem-like behavior. We can do serial transfers of tiny numbers of cells. Dirk Bush has shown you can even do it with a single cell and repopulate the whole host. So why are those two things characteristic of lymphocytes, tumor-infiltrating lymphocytes, and how can they simultaneously happen? So that's the talk today. So to understand this, I'd like to talk about how the balance of ions in the body. And this may seem far-fetched, but stay with me for a second. The inside of cells has got lots of potassium, about 145 millimole, and these are approximate values. But virtually all metazoan, maybe most life has this quality. The extracellular space has very low potassium, like 5 millimole, as we were describing, or even lower. So the observation is that when tumors die by apoptosis or necrosis, there are bags of potassium that are released into the tumor microenvironment. So this very talented fellow, this is the first time I've ever seen him in a tie, but Robert Isle published a paper a couple of years ago where he actually measured ion distortions in tumors, in mice tumors and in human tumors, and this is relative values of the ions. And there's lots of potassium in mouse and human tumors. And the other ions are not significantly altered. So how does this tiny, tiny extracellular ion have effects on T cells? Which were described in Robert's paper in Nature a couple of years ago. And that's the subject of today's talk. Okay, so how does a tiny ion that wouldn't even show up on this diagram, how can that affect epigenetics? And although this drawing is from a recent review in Nature Reviews Immunology, all of the data I'm going to show you today is unpublished. So to summarize, and I know it's after lunch, so for folks that may nod off, here's the talk. Here's the entire talk right here. The paradox of Till, how an ion can limit T cell function and preserve stemness. Okay. So one, high potassium acutely silences effector cells, and we'll go through that. It induces functional caloric restriction in lymphocytes. It triggers a functional program of calorie restriction, which in turn creates autophagy metabolically. this high potassium limits effector differentiation and preserves the memory-like epigenetics of cells and increases the persistence of T cells and their anti-tumor efficacy. So that's the talk in a nutshell. So this is a very simple experiment. Yeah, you can do in your own lab. If you just add physiologically relevance of high potassium, like 40 millimole, and of course we can address the osmolarity of that. We've done it both ways, just the addition of potassium or correcting potassium with sodium. T cells shut off. They stop making cytokines that we think are important. All right. So cells, why is that? So, I mean, we have tried to understand that more. Cells, as we've discussed, have an electrochemical gradient to facilitate the transport of nutrients. So let me analogize this electrochemical gradient to a plug, to the actual electrical gradient we use. So if you want to plug in your iPhone, you don't create a power plant to do that, or have a coal-fired, clean coal, of course. You don't create that power plant or nuclear reactor just to charge up your iPhone. You just plug it in. Well, cells also have a grid. They have an electrochemical grid, and they spend a lot of energy creating this grid across the membrane. And you can see this, that it's literally an electrical grid, and it's also a chemical grid. And you can use ions to transport substances against gradients. And so rather than having, a motor or something attached to every little action that you want to do, you could just use this electrochemical gradient to do things. And obviously neurons do that, but all cells do that. And so when we measured, we looked at the impact of hydrogen, of these potassium ions on the utilization of calories by the cell. So the experiment is very simple. You just take CD8 positive T cells and add potassium, and then quantify all of the metabolites that you see within the cell. And to make a very long story short, you can obviously do this using standard techniques, capillary electrophoresis coupled with triple quadruple mass spec, to read out what substances are there at a very high resolution. If you look at control, and this is just like 200 some metabolites, if you look at control versus cells grown in high potassium, all of the metabolites are depressed. I mean, this is just whatever is there, and we'll go through the individual metabolites on a case-by-case basis. And except there is this little band of metabolites that is not depressed. So let's look at that a little more. So the potassium does not compromise T cell growth. I told you it shuts off their ability to produce cytokines, but it doesn't shut off their ability to divide. And in fact, they don't die when you re-stimulate them. So a lot of times when we're doing this so-called repetitive stimulation, we see a dip in the cells. Here, you can see that the cells grow normally, and they die much less. So that's useful when you're trying to grow lymphocytes. So the functional caloric restriction is associated with reduced glycolysis. So let's look at these metabolites. So when glucose comes in, it goes to G6PD, and these other metabolites, and eventually to lactate. All of these substances, all of these, the glucose utilization pathway is depressed by potassium. If we just look at, if you add a fluorochrome to glucose, you could, this substance, 2-NBDG, is just fluorescinated glucose. You could see that potassium depresses the ability of lymphocytes to take up glucose. And here's the MFI plotted from several experiments. But having extra potassium in the extracellular fluid, which again kills this electrochemical gradient to some extent, it attenuates the ability of cells to take up lipids. Again, this is the same type of experiment where you use fluorescinated lipids and then look at the ability of T cells to take them up, and they take up less lipids. So the potassium conditioned cells have enriched. What are these enriched metabolites? They're part of a pathway I had never heard of before this, but it's many of you probably have. It's called the Kennedy pathway, and here are some of the metabolites associated with that pathway. And those are the metabolites that are elevated in cells grown in potassium. Okay, so what is the Kennedy pathway? Forgive me, this is sort of, might be tedious for some listeners, but this Kennedy pathway is a feed-in, helps create autophagy. So it creates the ability of the cell to eat itself, to utilize itself as a nutrient. And it does this through an orchestrated set of reactions that must have been very important during evolution. And I summarize here, and these are the metabolites I just showed you. And so it made us think, are these cells experiencing autophagy? Okay, so if we look at the impact of potassium on gene expression, so you know, this is very straightforward. You just take T cells, grow them in control media, regular media, or media with extra potassium. You profoundly affect the sequence, the RNA that's produced by that cell. And so we did this for all the genes using RNA-seq. Of course, RNA-seq just takes the RNA and then you make cDNA, sequence that, and measure the transcript. So what you get? So this is this great tool called gene set enrichment analysis, where you can look at all of the changes in the genome simultaneously by saying, so what genes are changed when you have potassium? And then you could compare that to other similar kinds of changes that are deposited by the Broad Institute and others, and then look at the comparison. So what do you get? Well, in this case, we saw this huge signature of autophagy, and it was very similar to cells that are incompetent. When we compared autophagy competent to incompetent cells, they had very similar changes to what we were seeing with potassium. So made us, again, think of autophagy. So how do we assess whether it's really autophagy? We can use fluorescinated components of these autophagosomes, and we can look at that Kennedy pathway to really read out whether or not, what the flux of this autophagy really is. And so that's what we did, and here are some of the constructs we used. And what we observed is that potassium does induce autophagy by this measure. So we can see this genetic signature of autophagy, but we can actually visualize it in living cells and look at these punctae. And of course, we're using genetically controlled situations where the component of the Kennedy pathway has a mutation, so it's not able to take up, it's not able to attach itself to the moieties because of a mutation here. And we don't see autophagy under those circumstances. So this, again, is just looking at all the cells using a FACS-based method, looking at the induction of this pathway in high potassium. So to summarize, potassium induces a functional caloric restriction. Why do we say caloric restriction? Well, the cells are bathing in calories, and they just don't use them. They don't take them up. What they're doing is using this so-called futile cycle, which is futile cycle, which has been dubbed that, but it's actually very efficient. They are engaging in autophagy, and we observe this high autophagic flux in cells cultured with potassium. Okay, so I mentioned earlier that this activity does not reduce their growth, and that's something we're struggling to understand. But it has a profound effect on their ability to utilize oxygen. This is referred to some as spare respiratory capacity, but the cells are actually more functional, more capable of inhaling and utilizing oxygen to do a very efficient process to generate ATP in their mitochondria under the conditions of hyperkalemia. So are the products of autophagy driving enhanced mitochondrial function? So when the cell is engaged in this process of starvation-like response, the cells are generating lots of things that can be taken up into mitochondria and then channeled into the Tca cycle we all learned about in college, if not high school. So this Tca cycle is very efficient at generating ATP. And the precursor, as you may remember, is this molecule acetyl-CoA, which feeds carbons into this metabolic cycle, which is extraordinarily efficiently at generating energy. So the crux of what I want to communicate is that there are two different pools of acetyl-CoA. So we were focusing on the utilization of this pool, but these two pools, the nucleocytosolic pool of acetyl-CoA and the mitochondrial pool are distinct. So they're generated distinctly by different enzymes, and it doesn't leak out of these mitochondria to go over here. This pool of acetylation is what acetylates proteins, including the histone molecules associated with DNA, and they can turn genes on and off in a heritable way. So cells can remember when they receive those signals associated with acetylation of genes. Okay, and that is going through so if this Tca cycle has lots of nutrients, it will spill out extra citrate, which can then be used in the generation of acetyl-CoA. So what are the levels of acetyl-CoA? So just to remind you, I'm going to propose that this starvation response triggers a very efficient creation of this cycle and energy generation and deprives the cell. So don't see export of citrate to create this pool of acetyl-CoA, and that affects the genes. Okay, and I want to try to tie that back into this tiny ion potassium. and how that affects the genes. All right, so functional caloric restriction depletes the nucleocytoplasmic citrate. So when we measure the total acetyl-CoA in this cell, most of that is associated with mitochondria. So these are all relative values. If we measure the cytoplasmic, the nucleocytoplasmic citrate in potassium, in those same cells, it's lower. And so the cytoplasmic citrate and its immediate precursor, the cytoplasmic citrate and cytoplasmic acetyl-CoA is lower in those cells. Okay, so to summarize, high levels of potassium in the culture augments oxygen consumption. The T cells conditioned in high levels of potassium are metabolically fit. And this is great because every hospital you can go get a bag of sterile potassium from the intensive care unit and even the floor. And so just by adding this potassium, you can make cells grow better and have better qualities. The high potassium depletes the nucleocytosolic acetyl-CoA, but there's more acetyl-CoA in the mitochondria. So if we zoom in, so you're probably skeptical if you're still with me. The conditioning of the cells so this experiment, we condition the cells in hyperkalemic conditions, and we see a silencing of the effector gene. So I'm going to show you that in more detail, but here's the paradigmatic effector gene, interferon gamma. If you look at this super enhancer here, you can see there's much less acetylation at that position. But before you look at that, take a look at the transcript. So this is just RNA-seq. So here you can look at the tracings in the transcripts coming off the DNA at any given location over the whole genome. And you can see here are the T cells making gamma interferon. These are the transcripts. And here's the transcripts coming off this DNA in hyperkalemia, which is much less. Okay, and we think that's controlled in part by this hypoacetylation. So the genes at H3K9, so that's a histone component, when they get acetylated, that's associated with turning the gene on. And we see less at this super enhancer and even within the gene body in cells grown in high potassium. We also see that the cells grown in high potassium have limited PD-1 mediated exhaustion markers. So there's less the potassium, you measure fewer H3K9 acetylation molecules at this particular site. And again, we can look at the whole genome, and I'll show you that in a second. All right, so I realize this might be tough to follow, but what I want to show with this slide is we're getting all of this off the entire genome, so we could look at the individual genes that are affected by this disruption of potassium by adding extra potassium, and it's the entire effector program. Here you can see, you can pick out favorite genes, and if we ever are able to publish this, you'll be able to look through the actual data. But having lots of potassium depresses the ability of these genetic changes associated with activation at H3K9. And here are all the genes that you may be interested in, granzymes, etc. So if we can integrate this all into a model, functional caloric restriction, we can see that under regular conditions, which are nutrient rich, you have this Tca, which is receiving carbons mainly through acetyl-CoA. But it's also able to spin off some citrate, which can be transported out of the mitochondria and used to generate this separate pool of acetyl-CoA, which is then used to activate these effector genes. But under high levels of potassium, this acetyl-CoA is higher, and this Tca cycle is not allowing citrate to go out, which is depressing the amounts of nucleocytoplasmic acetyl-CoA, which doesn't allow these genetic changes to activate the effector program. And it preserves a stemness-like phenotype, and I'll show you more of that. So to summarize, the high potassium depletes cytosolic acetyl-CoA and limits the acetylation of histones and the activation of effector genes. So the high potassium silences the whole effector, T-cell effector program. So so it shuts off the effector. So that's one side of the paradox, the tumor paradox. T cells in the tumor microenvironment are not functioning. They're not making an effector program. They're not making gamma interferon, but they're preserving stemness. So let me show you the data that we actually see that. I mean, so we can see that by transferring cells and looking at their stem-like qualities. And of course, we've done that. And we can use them to treat tumors better. But let's look at the genes here. So when we look at all the genes, this is just a simple RNA-seq experiment. If we look at the genes that we see these young stemness genes like IL-7, BCL2, BCL6, L-selectin, CD62. Well, these all mark the cells as being younger. And again, if we take all of the gene changes compared to normal versus high potassium, and we compare it in a gene set enrichment analysis, what is that like? That's like naive cells are to highly activated effector cells, KLRG1 positive cells, on a whole transcriptome level. So we can furthermore see that the ultimate stemness T-cell factor, TCF1, is robustly expressed when cells are grown under high potassium. And we can even see this just using a Western blot. There's lots of TCF when you grow cells in this Western using high potassium, and very little when you let cells go through the normal pathway. So if you condition cells in high potassium, you can preserve the memory-like phenotype of human cells also. So we work interchangeably in mouse and human. And so here's one of the gene therapies we've published on. So assigning T cells to have reactivity to ESO, and that is just a T cell receptor specific for ESO. Here's the gene engineered, and here you can see some markers in humans. CD27 and CD62L are preserved in cells grown in potassium. So they're the kinds of phenotypes we want to see. And here you can see, you don't see it in every patient, so here's 3 patients. One, we didn't see an appreciable increase in these memory stem-like markers. And humans, of course, we usually don't start reaching conclusions unless we've done 10. And here's just 3. So there's a lot more work to be done. So conditioning with the hyper-K also preserves neoantigen-specific T cells. So, we can, by genome sequencing, I'm not going to go too far into this, but by identifying the mutations that are present in the T cells, we can use that to identify the T cells that recognize those mutations, and the immune system is extraordinarily good at telling self from other. So mutated self from normal self. It's sort of its specialty. So if we fish out those lymphocytes and then measure the impact of potassium, we get phenotypes that we want to see. So specifically high CD62L. Again, not in every patient. Here are 4 different kinds of neoantigen-specific antigens, each one of them specific for a different mutation from different patients and different cancer histologies. But we see this general trend. All right, so usually we go from mouse to human, but and then to a clinical trial. But I think here we need to go from human to mouse, and that may seem regressive to some people, but doing a human clinical trial today is a lot of work with all the conditions you need for growing cells. And so let's try these maneuvers out in mice, realistic mouse models. So in mice, high potassium prevents the loss of the memory phenotype, which you see here, and it preserves this memory phenotype in mice. If you use cells conditioned in high potassium to treat tumors, here's a CD8 model, the PML model, developed by Willem Overwyk in our lab about 15 years ago, or this CD4 positive model developed by Pavel Moransky more recently. You could see you could improve both of those kinds of cells. And so we're adoptively transferring these to mice bearing large established tumors. Usually those tumors have been growing and they're vascularized for at least 10 days before we treat the mice. And you can see you can get these fantastic improvements. So here you want to be low. So you want very little tumor growth or tumor regression in those cells. So cells grown in high potassium improves cells even when you normalize the number of cells you're giving. They're better, they're qualitatively better, and they also survive longer. I'm not going to show you that in mice. So they're more stem-like. So to summarize this part, ex vivo exposure to high potassium maintains CD8 positive T cell stemness. It preserves their phenotypic traits of human TILs for effective immunotherapy, and it does the same thing in mice. And high potassium enables the generation of anti-tumor T cells capable of enhanced tumor treatment in mice. So we haven't done this in humans yet. Although it's very doable, since the potassium is not like a pharmacological reagent. And we've had some trouble getting pharmacologic reagents, as everyone has. All right. So let me just take you a couple of more steps into this, and I'll try to keep it short. So if you have any questions, I can answer them, attempt to answer them. So acetate is a key precursor for this acetyl-CoA. So the acetate helps create this acetyl-CoA and feeds into this Tca cycle to make this energy-generating cycle go better. And then, of course, if there's excess nutrients, you get export of the citrate and creation of the nucleocytosolic acetyl-CoA pool. So if we provide external acetate, we can undo a lot of what's happening. So here's two markers, the CD44 and CD62L. I told you about this, the memory-like qualities of these cells. But if we give extra acetate, they look just like control, even though they're in the presence of extra potassium. So the external acetyl-CoA provisional of this also affects. So if we give acetyl-CoA itself. Okay, and we push that into cells, we can undo a lot of what potassium is doing. So here, gamma interferon, the ultimate effector gene, is reduced under conditions of high potassium. But if we add acetyl-CoA, which could build up those, that nucleocytosolic program, we can now acetylate these genes and activate the effector program. And so this shows that even in the presence of high potassium. The external acetate normalizes the actual epigenetic status. If we look at the H3K9 chip, that's just chromatin immunoprecipitation, you can see for the gamma interferon promoter that potassium, as I showed you earlier, is reduced under condition, reduces this acetylation. But if you add acetate, it's increased. Or same thing for the PD-1 location. So pharmacologically, so we wanted to pharmacologically mimic the reduction of the nucleocytoplasmic acetyl-CoA. And one key step in that creation of this nucleocytoplasmic pool is this enzyme, ACLY, which can be pharmacologically blocked with several substances, but including an altered form of citrate, which just jams that enzyme, and so you don't get creation of this pool. So the 2-HC depletes the cytoplasmic acetyl-CoA. So it actually works, and the histone acetylation at the interferon gamma promoter. So now here we're not using potassium at all in this, we're just adding 2-HC, another substance we could use to grow T cells better, and seeing that this does the same thing, it limits the effector program, and to make a long story short, it preserves the stemness program. All right, so the calorie restriction mimetic. So this makes cells think they're starving because there's no acetyl-CoA, even though there are nutrients. Look at the impact of 2-HC, just added to the media. You can see this preserved stemness phenotype with more CD62L and less gamma interferon. Again, this is just by affecting the generation of that acetyl-CoA pool. So the 2-hydroxycitrate also triggers the same pathway, autophagy, and reduce acetylation of the effector gene. So let's take a look at that quickly. Conditioning with 2-HC improves the anti-tumor efficacy of tumor, and these tumor curves we're getting are probably even better than those we get with potassium. You can see, again, you want to have no tumor left here, and you can see starting off with large tumors, so these are 1 centimeter across. You can see these cells are working better, just adding this metabolite that jams the generation of this nucleocytosolic pool. And the mice survive. So preserved stemness following high potassium requires depletion of nucleocytosolic acetyl-CoA. Restoration of acetyl-CoA reverts the high potassium phenotype. 2-HC results in enhanced persistence in anti-tumor clearance and host survival following adoptive transfer. And I'm not showing you each and every piece of that, but I wanted to get through. And finally, I just want to provide some genetic evidence. Some people don't believe these, experiments adding substances to T cells. I want to see genetic experiments. Okay, so here's some genetic evidence. So let's look at these enzymes that actually generate the mitochondrial and the nucleocytosolic pool. And those are 2 different enzymes that are similar to each other, but they're different in their localization and their function. This one's in the mitochondria, okay? So if we look at this starvation-induced condition, what do we see? We don't see much of a change in this ACSS2, but we do see a big change in ACSS1. Okay, so the ACSS1 is induced. So this is not an accident. So this is a coordinated cellular event associated with starvation. And look at the ACSS1. So if we just take and make a retrovirus encoding this gene and force cells to express ACSS1. Again, this is not using any kind of potassium or affecting anything else in the pathway. You just either use a retrovirus to put in ACSS1, another one to put in ACSS2, or a control retrovirus. You see this increased oxidative capacity using ACSS1, which we think is facilitating that mitochondrial event. And if you force cells to overexpress ACSS1, you improve T-cell-mediated tumor clearance. And here's that experiment. So this is a purely genetic experiment, treating cells with a control retrovirus engineered cells or with the ACSS1 engineered cells. So to summarize, high potassium-induced caloric restriction increases ACSS1 expression. Overexpression of ACSS1 augments oxygen consumption and improves the anti-tumor efficacy following adoptive T-cell transfer. So to conclude, a local ion with a van der Waals radius of 2 angstroms can profoundly affect T-cell function. A tumor-associated hyperkalemia and possibly other conditions where you don't want a lot of inflammation, like frostbite or crush injury. These potassium ions suppress the effector function, and they preserve the stemness function of T cells. And high levels of potassium accomplishes both components of the tumor paradox, the tumor infiltrating lymphocyte paradox. It suppresses the function, the effector function, and preserves the stemness, and it does this through cell metabolism and epigenetics. So high potassium-induced caloric restriction increases ACSS1. Overexpression of just this enzyme augments oxygen consumption and improves cells after adoptive transfer. And so I'd like to end there thanking all the members of my lab, especially Suman Vandala, who did a lot of this work, and Robert Isle, who's back at his surgical residency and heading at Oregon, heading to Sloan Kettering in the fall. Talented surgeon. Started much of this potassium work using his insight as a really a neural, into the ways neuron signal. And so we also had lots of help from many that are not even listed on this slide. And I would finally like to thank so many members of the clinical team spearheaded by Steve Rosenberg, which has many talented physicians. We wouldn't be able to get these human samples without talented surgeons, which help us a lot. So I'll end there and take any questions. Thank you.
So I think we have time for a couple of questions. Maybe while anyone's coming up, I can ask you, how long do you need to have potassium in the culture to get these effects? What happens if it's there for the initial part and then you take it out?
If you take out the potassium, so it's entirely reversible.
Right.
So the impact of potassium is there when the potassium is there. And when you take it away, and we're focusing on these reversible inhibitors like the 2,8-C, is also reversible. The cells are free to go on their way and to develop effector function. Okay. And I should note that that's really important because we need the cells to become effectors. So we don't want them to stay like babies or stem cells the whole time. We want them to grow up and go out there and kill. We just don't want that to happen too soon.
Okay.
Thank you. Does it work? Yes, great talk. So potassium increases the stemness of T cells as long as you have a high potassium in an extracellular.
I'm not hearing. Maybe if I stand over here.
Yeah.
Go ahead.
Okay. So you have high potassium and you increase the stemness of your T cells. These T cells then enter the tumor microenvironment and they do find a lot of potassium. How do they turn on the effector potential if they still find a lot of potassium there? How do they revert back to effectors? Thank you.
Yes. So this is, okay, so this gets into some highly contested areas. So most immunologists, T-cell immunologists, will tell you that effector cells can get reprogrammed back into stem-like cells. We don't measure that. We never have. It's a linear pathway of differentiation. So once cells become effector, they don't go backward. They retain their effectiveness. So when they go back into that environment, and this is somewhat speculative, but we don't measure the cells going back into a stemness state or shutting off their effector genes. And they already have these acetylated genes that are activated as part of the effector program. So we're not, and we can't give extra potassium to an organism without killing it because of cardiac complications. And so that's really a speculative answer to your question.
Thank you.
Okay, Marcella.
Nick, that's a great talk. Takes us into a new direction. I'm just thinking, did you look at the role of the insulin receptor and how much insulin receptors on the T cells? Because as you know, insulin controls the influx of potassium into various cell types and how that
Great
might be
question.
affected.
Yeah, we haven't looked at that, but I mean, all of the gene products are there in the RNA-seq data. It would be interesting to look at that.
Thank you. That was a fascinating lecture. I wonder if you've looked at the effects of high potassium on glutamine uptake, because you might argue that anaplerotic metabolism could do some of those things to acetyl coenzyme A.
One more time, please.
Glutamine? Have you looked at glutamine uptake?
Glutamine uptake. Sorry, that's mine. No, we haven't specifically looked at glutamine, but it would be interesting since that's an alternative.
I think it would be
fuel
really
source.
interesting. Yeah.
But I would speculate wildly that cells wouldn't utilize extra glutamine, but they might. I don't know. We just measured glucose.
Are you a betting man?
Okay, one last question.
Cool.
Does your ACSS1, does it change mitochondrial membrane depolarization? Because you have a big change in your oxygen consumption, like FCCP?
Yeah, that's simple to measure, but we haven't done that. That's an excellent experiment.
Okay. So thank you very much, Nick, for a very provocative and outstanding lecture.